This talk discusses the fetal adaptation to a low PO2, essentially why the fetus doesn't just die. Once again, I want you to remind yourself of the fetal circulation. I follow a blood cell through and understand how the oxygenation changes a bit, not in detail, but in general, how that oxygenation changes with the least oxygenated blood arriving into the umbilical arteries that then feed the placenta. Oxygenation occurs, nutrient exchange occurs, waste product is liberated, and fully cleaned and oxygenated blood leaves through the umbilical vein, which joins the circulation in the liver, and the portal circulation at one point gives off a vessel, a shunt called ductus venosus, that enters directly into the right atrium, not the way it's described in your embryology diagrams. Another objective is to contrast the characteristics of fetal and adult blood that do affect the oxygen saturation curve, and then describe how fetal hemoglobin can accomplish this high affinity uptake across the placenta and a lower efficiency, high delivery of oxygen at the tissue level. Under what circumstances does that occur? And of course you remember that there's no maternal fetal blood mixing. The maternal blood that enters the placenta through the uterine arteries surrounds that placental tissue with the highest possible oxygen. It has a PO2 of 100, let's say. Whereas the deoxygenated blood that comes in from the umbilical arteries and enters into those vascular spaces in the placenta that are apposed to the vascular spaces that the maternal blood enters into, leaves a membrane in between. And the difference in those two PO2s, of course, encourage that transfer of oxygen. However, the placenta is structured so that there's a general equilibration of that blood. The fetal blood is never becomes as highly oxygenated as the entering maternal blood. And you're left with a PO2 of about 35 to 40. That's the highest PO2 that a fetus is going to get. The highest PO2 is going to be then drained from that placenta into the umbilical vein, and that blood is then at some point going to become less and less oxygenated as it courses through the fetus. So remember, the fetal blood at its highest can't get any higher than 35 or 40 PO2. This is a diagram of the fetal pH compared to neonatal, or us. Fetal PO2 compared to neonatal or adult. But surprisingly, the saturation of the fetus, despite its low PO2, is up to 70%, not as much as us. PCO2 is quickly exchanged across the placental membrane. And importantly, the base excess is relatively normal. So even though the saturation is low and the PO2 is really low, the base excess shows that this fetus is not producing products of anaerobic metabolism. It's not dumping acid into its circulation. And of course, the fetus doesn't have any extra protection against that. It has the same kind of tissues and metabolism that adults have. And so somehow, that fetus has to be at the cellular level working in an anaerobic working in an environment that's aerobic. It is not an anaerobic environment. It is not a different kind of organism as a fetus. It just has adaptations that allows it to deliver enough oxygen per gram of tissue at the cellular level to support aerobic metabolism, just like us. So the first solution is the characteristic of fetal hemoglobin. It has a higher, and of course the fetus, it produces a great quantity of fetal hemoglobin compared to what we have in adults. And the fetal hemoglobin has a higher affinity for oxygen, such that at a low PO2, a saturation is higher than it is in adult life. And that we call a leftward shift on the PO2 and percent saturation curve. You can see here the saturation curve. This is the PO2, partial pressure of oxygen. And here is the saturation of fetal hemoglobin. And you can see that at a low PO2, the fetal saturation is actually much higher than adult would be. Adult saturation would be here, fetal saturation would be here, quite a big difference. And that occurs more at the lower ends of the PO2, to the degree that this fetus is capable of maintaining a saturation level of a maximum of about 70. Plus, there's more fetal hemoglobin because the fetus has a higher hematocrit as a normal circumstance, normal situation. And another thing, there is a component of the blood, 2,3-DPG, or sometimes it's called 2,3-bis phosphoglycerate. And the presence in cells decreases affinity for O2. So 2,3-DPG decreases affinity for O2. And the fetus, who's clamoring for oxygen, as I said here, produces a hemoglobin that can't interact with the 2,3-DPG. So the fetus then isn't inhibited like the adult hemoglobin might be. And so that shifts that saturation curve also to the left. So it grabs more oxygen at less PO2. It grabs more oxygen because it has a less of an interaction with the 2,3-DPG, which would inhibit that O2 uptake. And remember, in the adult, after fetal hemoglobin declines, 2,3-DPG interacts with that hemoglobin A, and its action decreases the affinity of adult for hemoglobin for oxygen. This diagram is an old diagram, but it says exactly what we need to see here. This is a saturation curve again. This just shows a fetus or an adult here. And what we mean by a leftward shift are all those things that increase oxygen affinity. So pH that goes up, temperature that goes down, CO2 that goes down. Under these circumstances, the ability of that hemoglobin to grab oxygen is diminished, and here are the conditions. In particular, as pH goes down, temperature going up, fever, acidosis sometimes from sepsis, PCO2 from poor circulation. So some of the untoward effects that occur in fetal life can actually inhibit its ability to oxygenate, let alone directly impact on its oxygenation for reasons of circulation or insufficiency of the placenta. And another thing, fetal cardiac output is very high per gram of tissue. And it's twice the heart rate of the adult. That's the reason why, based on that equation of cardiac output times stroke volume and heart rate. Stroke volume is pretty constant. Remember, the fetus operates at a high volume content of the heart. The fetus does operate at the top of that Starling curve, and so it can't react to increased pressure on that stretch receptors. It can't react to increasing blood volume in the heart in the usual way by increasing its contractility. And so its only response often to conditions that are adverse is to become tachycardic in order to increase its stroke volume. Stroke volume is one of the ways that very high stroke volume is one of the ways that a fetus supplies that peripheral tissue at the appropriate oxygen molecule per gram of tissue to maintain aerobic metabolism. Shunts are interesting because shunts in the fetus prevent sort of wasting of the circulation into areas that the fetus really does not have to oxygenate as highly. For example, the lungs. There's a shunt through the ductus arteriosus away from the lungs. There's no problem with that because there's enough oxygen per gram of tissue to keep the lungs alive. There's no need to put more blood in there. There's no oxygen exchange going on. Also, what's very interesting is that the shunting contributes to a lower organ tissue pH. And remember on the saturation curve, as the pH goes down, the affinity of that hemoglobin for oxygen goes down. So at the tissue level, delivery is actually increased by that oxygenation decreasing, by that pH decreasing as the fetus begins to rev up a little bit of anaerobic metabolism. And finally, fetal polycythemia, meaning lots of red cells, high hematocrit, makes the fetus able to deliver more blood cells in the same amount of mils of blood to that gram of tissue that it needs to oxygenate. Now remember, there's a rapid equilibrium of CO2 across the placenta. And at this point, the idea is to have a higher pH than that is present in the tissues. And remember, on that oxygenation curve, there's a shift to the left as the pH goes up. That, again, allows the fetus to grab more oxygen when it has the opportunity, and based on the lower tissue pH levels, deliver that oxygen when it needs to deliver it to the tissues that are peripherally. And the result overall is to keep that fetus a regular aerobic organism just like us, without evidence of acidosis. Thanks.